everybody. Sorry for the small technical difficulties here. I think we're now go. We're gonna, because of that, we're gonna go with this session to 3:45. Hopefully join for the whole time. It is a real honor to share this session that's dedicated to the 20th anniversary of RNA interference in mammalian cells. Obviously, a mark in the entire field of RNA interference, but also discovery for the benefit of discovery of RNAi therapeutics, something that I care a little bit about as the CEO. But most importantly, we have some wonderful here joining me, Craig Mello, Limor Joshua Tor, and I'll introduce for each of their talks. But I want to highlight the fact that we also have some on-demand talks with a number of speakers who have been selected to session. These include Omer Weisbrod at 11. He'll be talking about sRNA screens, CoV-2. Yvonne Longjean, Leiden University Medical Center. She'll be speaking about specific silencing of von Willebrand's factor. And Eric Wickstrom at Thomas Jefferson talking about imaging the Huntington RNA for diagnosis and therapy. Certainly a very talk. All three of those are important talks. So let me now introduce somebody who doesn't need any introduction, and that's Craig. As you all know, is a professor of molecular medicine at the University of Massachusetts Medical School, just down the street from those of us here. He was awarded the 2006 Nobel Prize for Physiology, along with Andrew Fire, for their joint discovery of interference. So this is where it all began with their seminal work in 1998. Craig is also an HHMI leader since 2000. So without further ado, I hand it over to you.
Thanks, John. Am I live? Great. Well, it's great to be here. Sorry again for the delay. We had some technical problems. So it's great to be here, and what an exciting time, oligonucleotide therapeutics. I'm going about something new that's fairly preliminary is more phenomenological rather than at this point. And that is a story to how organisms license information. That recognize self versus non-self information, at least in the Oligons. I'm not sure. There we go. And this is just a nice thing. The ovary with the eggs maturing. I love to say this. If you've heard me talk before, it's just something that really can't say enough. We always underestimate living things. They are just sophisticated. The germline is an issue as it relates to information. The germline captures information. You might even think of the timescales like an antenna for information, bringing in new information from other genomes that it encounters, of course, from viruses. And then once that information, it can be silenced, it can be shuffled, and it can be so that it can be expressed later. Without that flow of information, there would be no humans, probably some slime on the planet. Anyway, here's the worm, and this is the real Nobel Prize winner, as I also like to say. And this organism just has a whole lot still to teach us. When Andy Fire and I started injecting into this animal, we were trying to essentially develop a new type of tool for animals. That is the ability to reintroduce genetic information. It had never been done in an animal at the time that we both started. And it turned out that the organism, perhaps unsurprising to us now, was responding to that information and, in fact, silencing it. And it was somewhat vexing, especially for Andy, because Andy had chosen a DNA vector that expresses, it needs to be expressed in the germline to confer a phenotype. And in order to deliver that genetic information, he found that he had to inject into these germ nuclei, which was technically very difficult. Fortunately, I was sort of in parallel trying to develop the same technique, and I chose a marker that's expressed in the hypodermis of the somatic tissues of the animal. And this turned out to be very fortunate indeed, because I could inject into this syncytium here a large extrachromosomal array rather than integrated DNA would then form and stably express the DNA, causing a phenotype that was easy to score. So we really, together, Andy and I eventually really got this organism going in terms of ability to transfer in DNA. But this vexing difference, this problem, the germline was really difficult to introduce genes into. The germline was just very careful, apparently, about allowing us to drive gene expression. And this is where the story gets interesting, and I'm not going to go into all the detail. But again, organisms are just so fascinating. This is a movie of a single-celled animal. And these are ciliated protozoans, and they're really huge cells. This is a metazoan here crawling in the background. You'll see another one in a moment. But here you have a single cell that's as large as a worm. This is another nematode-related species. It's not a nematode, but anyway, it's suffice it to say that in order to be large, these cells, and another amazing thing about them is a cell that's capable of coordinated movement. It's sending, coordinating the motion of all the cilia in order to capture food and to swim in an environment. Just again, you cannot imagine sort of the complexity of a single-celled animal. So how do you get to be this large as a single cell? How do you make sufficient genetic information? Well, in this organism, you expand the genome many thousand times by producing what's called a macronucleus. And in order to do that, the genes in the genomic DNA, the germline DNA, are actually excised from the chromosome into short genes. And in the process that's guided by Argonauts and by RNA, the genetic information is actually unscrambled. The DNA itself is spliced to put the gene together. So again, amazing, really what Argonauts can do, the guided genome reorganization. So Argonauts can function to modify genomic information. Another thing, just to sort of give you a scope of sort of what nature is telling us is possible, this is a image of a planarian where you can see the cells called neoblasts in the top panel. These are the cells that are expressed, that express a germline, an Argonaut that's also prominent in the germline called a peewee Argonaut. And these cells are able to respond to injury, taking cues from the somatic tissues to regrow the entire missing part. In fact, if you cut off the head, it'll grow back the posterior and vice versa. Imagine if humans could grow back heads. I know some humans who would benefit from that personally. So again, realizing the promise of informational drugs requires digging into and understanding how organisms handle information, in particular foreign information. When foreign information enters a cell has multiple options. It can, of course, kill itself to prevent the information from spreading. It can destroy the information, or it can silence the information. Argonauts, especially in metazoans, are clearly functioning to silence information, which I think is a really interesting tool for an organism, especially the germline, to use in order to essentially enable the organism to do the following. That is to take that foreign information, shuffle it into the genome over evolutionary time, and then potentially relicense it to express it later. For those of you who haven't thought about this, just consider the mitochondrial genome, which has mostly migrated into the nuclear genome. And all of that rewiring has happened over the course of evolution, such that now the nuclear genome flawlessly expresses those formerly microbial genes. Just absolutely amazing what organisms can do with information. So in that theme, I just want to take you through some of the things that happened in the course of our investigation of RNA interference. So it turns out that the germline is carrying RNA memories on board its Argonauts. There are Argonauts expressed in the germline. These are pictures of the ovary that are stained with either an antibody or with GFP detecting 3 different Argonauts. The Caesar-1 Argonaut engages small RNAs that are made by an RNA-dependent RNA polymerase templating from all of the expressed mRNAs in the germline. Basically providing a memory of self. This Argonaut and its guides are passed in the sperm and the egg to the offspring. A second Argonaut, the Wago Argonaut family, there's actually multiple Argonauts in this group, are also present in the sperm and the egg, and they are transferring a memory of genes that are undergoing silencing, if you will, non-self, although they're part of the genome and could be expressed later, so I don't think that's really a fair way to describe them. But genes that are being silenced, a memory of the silencing state is being transferred by the small RNAs engaged with this Argonaut. And then a third Argonaut, the Peewee Argonaut, which is also the one expressed in the neoblasts of the planarian, is present in the germline and we believe functions to scan RNAs looking for foreign RNA that is not previously recognized by Caesar-1. So again, incredibly complicated model, but apparently the organism is, rather than remembering foreign things, which it also does, it also remembers its own gene expression. And that enables it to innately recognize foreign genes because they come without that memory of self. Anyway, the story began for us really after the discovery that double-stranded RNA could trigger this phenomenon with a genetic screen to try to find genes that are required for it. And the screen itself is shown on the left. It was very straightforward, really. We mutagenized animals, exposed them to RNAi by feeding, believe it or not, that would enable the worms that ingest the bacteria expressing a double-stranded RNA to produce an effect, in this case, silencing an essential gene, causing the embryos to die. Hiroaki came up with this screen and did it and found lots of mutants that were defective in RNAi because they were now able to produce viable progeny, as shown on the left there. And the mutants he discovered immediately hinted at this complexity. And it was really, I felt, incredibly exciting because he found 2 kinds of mutants. He found mutants that were completely defective in the response to double-stranded RNA, and he found a second group of mutants that were mobilizing transposons. So in one group, they were not responding to the double-stranded RNA anymore, but the transposons remained silent, which made us believe that there must be 2 modes of silencing or triggers, 2 different triggers, one double-stranded RNA and something else that was triggering transposon silencing. And I already hinted at that, and I'm not going to talk about it, but it turns out that the Peewee Argonaut system plays a role in that. And we think on an evolutionary timescale, when new transposons are encountered, the Peewee system recognizes them and establishes silencing. And this is done by virtue of a Pol2 expressed guide RNA. Peewee Argonauts engage RNAs that are not processed from double-stranded RNA, but rather from transcripts made by Pol2. So very, very different triggering mechanism. So I'm always told, you need to simplify your models because my models are incredibly complicated. This is just how RNAi was explained by CBS News back around 2006. You can see the nice short double-stranded RNA, and there are the defective genes. Very, very nice model. Unfortunately, life is not quite that simple. So here again, just to summarize some of the complexity that's going on, and this again is just the beginning. There's a memory of self-gene expression. All of this is produced by RDRP templating from mRNAs, and there's a memory of genes that are undergoing silencing. Those are passed on in the gametes. We think when a new transposon, virus, what have you, is encountered in the germline, the Peewee Argonauts, which exist with multiple different guides, tens of thousands of guides are encoded in the genome, can engage those guides, search out, find the RNAs that are not targeted by Caesar-1 and initiate silencing. Okay, so by way of background, this is the sort of the layers of complexity that already exist. And so we were experimenting with, Cas9, and like everyone, we wanted to have Cas9 expressed in the germline. And it turned out that Cas9 was silenced. This is not surprising. However, when we mutated genes involved in RNA interference, the genes remain silenced. The Cas9 transgenes remain silenced. And it turned out that other groups were able to show that this was in part due to intron insufficiency. The Cas9s that we and others had constructed just didn't have enough introns in them, it seemed, or the introns were not of high enough quality, and the genes were not being expressed in the germline. They were expressed in the soma, as you can see in the L1 and L2. This construct is abundantly expressed in the hypodermis and the gut, but it's completely absent in the dissected gonad in the lower right panel. So we went back based on this. This was both frustrating, but anytime a phenomenon like this crops up, it's also an opportunity to explore it. And so we set about a whole project to try to understand how introns are engaged by the silencing machinery. And this is still very much a work in progress, although we published the first paper more or less describing the phenomenon just recently in Developmental Cell, Makeyeva et al. 2021. It turns out that if you take the introns out of an endogenous gene by CRISPR, it becomes a default target for RDRP. RDRP, it seems, I should also note, as I said earlier, is engaged with all RNAs, including those that are expressed. But when there's no introns, it seems by default, the RDRP loads the WAGO pathway. I should see that's shown here. When there's an intronless gene, Peewee scanning is not it's totally irrelevant. RDRP scans intronless genes and loads WAGO Argonauts directly without the need for Peewee, which has led us to sort of refine this very complicated model involving Peewee as a scanning factor. We think that by default, these RDRPs are loading Caesar-1 when introns are present, and that Peewee can override that if there are high affinity target sites. PiRNAs engage their targets much as microRNAs do, which means they use seed pairing. And the better the pairing, sort of the higher likelihood that they'll recruit the silencing machinery. So again, very complicated, and it seems tied in some way to mRNA splicing. And this is purely phenomenological at this point, but now we know that at least these RDRP systems are somehow sensing splicing, and we don't know how yet. So that will be an interesting subject for further study. Takao Ishidati in the lab decided to do a forward genetic screen to try to better understand intron insufficiency. And when he did the screen, he started with the Cas9 that was silent, and he did a screen, and he found a lot of genes that turned on Cas9 activity. And the screen was very straightforward because he started with guide genes that were expressed but not functional because the Cas9 itself was not being made, and then looked for cutting and was able to find quite a few mutants. But what he found was that mutants that disarmed the small RNA pathway activated the Cas9, but only in the mitotic zone. The activation did not spread into the pachytene zone of the germline. And yet, it seemed that mRNA was being expressed in the pachytene zone, as you can see in the mRNA fish image. So the knocking out the small RNA machinery partially activated the Cas9 transgene, but only in the distal region, not in the more sort of meiotic region of the gonad. And the mRNA signal was completely absent in wild type, suggesting that silencing was at the mRNA level, and that there was a second mode of silencing that was not at the mRNA level, if you will, in the pachytene zone. And so this is just a close-up showing that the RNA there in red is visible. So Takao set out to do an additional screen starting with the NERD2 mutant background. This is a mutant defective in a nuclear branch of the small RNA silencing pathway that, as you saw there, activates Cas9 distally in the mitotic zone, but not in the proximal region. And then he did an RNAi screen looking for mutants that activate the gene in more proximally in the pachytene zone. And so he found a whole bunch of mutants that do this, and surprisingly, they are components of the proteasome. Now, this was interesting, but a little worrisome, because you could imagine that maybe protein expression distally was sufficient if the protein's not turned over. And so maybe there's really no change in gene expression, just persistence of the protein. So we weren't really sure what to make of this, although it sure looked like, as you can see in this image, even the oocytes are now abundantly expressing the Cas9 protein. So in order to further investigate this phenomenon to see if it's really a proteasome-mediated silencing mechanism, we use this viral sequence called a T2a sequence. This is a sequence that some viruses, a strategy that some viruses uses to get the ribosome to release the upstream polypeptide and then essentially start, continue elongating, making the second polypeptide. And the reasoning here was that the GFP part has adequate splicing. It has the normal complement of introns. And we thought perhaps if we separate these two domains, if there's something recruiting the proteasome co-translationally, if you will, to the Cas9 part of the message, that maybe this would enable expression. And amazingly, this worked. You can see that in wild type, Cas9 with the T2a GFP is completely silenced. But if you now inactivate one of these small RNA pathway genes, the Cas9 GFP turns on throughout both the distal region and the pachytene and oocyte region of the gonad. So remarkably activating sort of this mRNA that we know is there for translation. It's just that the proteasome normally is destroying the polypeptide seemingly co-translationally. So there are layers. The point here is that there are multiple layers at which the organism is silencing this gene, and there must be multiple modes of licensing required, I think would be the inference for normal gene expression. Again, the small RNA pathway keeps the gene off, and if you remove the small RNA pathway, there's a co-translational destruction of the RNA that has insufficient. Looks like my model slide is absent. Oh, too bad. Well, I've already, well, the model slide just shows that there are multiple layers here of regulation. There's a small RNA dependent mechanism that also chromatin silencing. And then additionally, there seems to be a layer that is small RNA independent that is also induced by somehow triggered by splicing. And the mRNAs that lack those licensing features that are deposited co-transcriptionally, presumably during splicing, are then recognized by the ribosome and degraded. The protein products are degraded co-translationally. So again, we don't know the mechanism. We don't understand how this works. Presumably, there are proteins that associate with the mRNA at the UTRs, perhaps, that establish or link this proteasome silencing to the RNA in some way. Again, really, we don't understand, and I think it's going to be fun. But the point is, there are multiple modes of silencing, multiple layers of silencing, and I think that organisms have these layers because, information is incredibly dangerous, but of course, it's also potentially very useful. So if you can handle it safely, it's probably better on an evolutionary timescale to keep information than it is to simply destroy it. And I'll end to take any questions and thank you.
Thank you, Craig. It was wonderful, and I completely agree that the wonderful beauty of life is just incredible, and your talk that so poignantly every time you speak. I was going to ask you, for new heads, but I thought I would avoid that question. But I really want to push a little bit more on the discovery of in germline cells, and you seem to suggest that it was a mechanism to better enable detection of foreign RNA. Do you think there's more to it than that? Good.
Yeah, I mean, this is also true, of course, in this related there briefly, but the ciliates have a equally remarkable genome RNA guided memory. So they basically are passing the sort of the key to unlock the genome is passed in the form of RNA. So I think it's a lesson for all of us to think about how RNA can guide the regulation of not only the expression of the gene, but also the arrangement of the genome itself. Yeah. And so there's a lot of lessons, I think, still to be learned. And this is why, the basic science, I can't plug it enough. I think the future of oligonucleotide therapy and other informational therapies is truly exciting. Yeah. Just seeing how far we've come, with, so rapidly, for example, with CRISPR and also the huge promise of Argonaut mediated gene regulation. I think there's just really, really exciting opportunities.
Yeah, absolutely. Well, Craig, I think in the we should move on, but I again want to thank you for that inspiring by all accounts. So we're now going to move on to the second speaker, Limor Joshua Tor, who is an HHMI educator and also the W.M. Keck Professor of Structural Biology at Culture. Limor's lab studies the molecular basis of nucleic acid regulatory, RNA interference, and DNA replication in particular. She discovered the enigmatic protein called Argonaut at the heart of the RNAi machinery. And of course, this has been the subject of her wonderful work that she'll now share with all of us. So, Limor, please take it away.
Hi, thank you so much, John, and thank you for having me for this wonderful celebration of RNA interference. And I can't believe how many years it's been. So I'll take you a little bit to some of our journey with the Argonauts. And of course, I don't mean Jason and the Argonauts. I mean these Argonauts, and I just wanted to start by saying, sort of following what Craig was saying, that there are a lot of pathways that we knew about started becoming connected through these RNA triggers, whether double-stranded RNA and also single-stranded RNA. But then we really recognized connected, not necessarily because of the RNA, but really because they shared certain proteins that were responsible for how these pathways actually worked. So you all know that one of these is dicer that's involved in biogenesis of some of these small taking either microRNAs or precursors of microRNAs or precursors of siRNAs, processing these into small bits of RNA and then loading this into a complex that was named RISC, the RNA silencing complex. The single strand that remained bound to this complex was called the guide RNA, and then that directed this to a target RNA, and when there was perfect complementarity, it caused an endonucleolytic cut, which we called slicing, or when it wasn't fully complementary, it was silenced in other ways. I about this at the earlier days of RNA interference. What really was how many different scientists of biologists came together to really how this thing works. So geneticists, biochemists, and bioinformaticists, and also lots of different organisms. You just heard the really groundbreaking work in worms and in very early days of the fact that the RNAs were really small, and again, microRNAs in worms and flies, and then of course we're the discovery in mammalian systems. So we thought this was really fascinating and thought that we can bring our angle of biology and also some biochemistry into this. As I said, that there was one important player that was recognized early on, that was dicer. And here we already recognize that it was a nuclease. It has at least two domains that were recognizable as RNase 3 domains, and then it also helicase domains, and others may be less recognizable. But there was another protein that seems to be around always, and Craig just talked a lot about those, and these are the Argonauts. These were more mysterious because we knew it had two domains, a PAS domain and a PIWI domain, but we didn't know what they looked like. They didn't look recognizable. They seemed to be around 4 RNAi type proteins, but that's about it. So we decided to go after this mysterious protein again. And in fact, now it really almost defines a process being an RNAi related process. We tried to figure out what it was all about, and a graduate student at the time, Jijun Song, started by looking at the PAS domain of fly, got a crystal structure of this PAS domain, and then using some biochemistry showed that the PAS domain binds 3' ends of RNA, and was doing so through this cleft of this domain. We figured where it was binding and what it was binding, but we were completely wrong in how it's. That came from work by Dinsha Patel, which showed that looking, binding very, very tightly to this phosphate between the two bases at the end of the. So Jejun was very, very insistent in trying to get really the full picture of an Argonaut, and I was trying together with Stephanie Smith, who was a technician at the lab at the time. This is who was a graduate student. She is now my lab manager. She stuck on with me. Decided to blast the database Peewee domain and found a protein heel from Pyrococcus furiosus. Pyrococcus furiosus ball. This is why we have this little ball here. And it had the Peewee domain very clearly by sequence, and it was about, but we couldn't really see a PAS domain. Jejun got crystals of this, very happy, and then even more happy when he got the structure. And this is what of this Argonaut from Pyrococcus furiosus looks like. We realized that there's two other domains we called, and this green one here, the mid domain. I'm sorry, it was probably too many hours in front of the graphic. We weren't more imaginative with our naming. And then there was a couple linkers that were connecting these domains together. We were to see this red domain, the PAS domain here, because we weren't sure it was going to be there. The sequence doesn't align very well unless you can use the structure to align it. But you can see that the structures align really nicely. And what's really cute was that this side chain of the histidine was really the same in the same location in space, though it was coming from different parts of the protein. But of course, the real surprise here was the Peewee domain. And it was kind of interesting because right before that, I was helping make figures for this textbook, Molecular Biology of the Gene, at the time after a big break. And I made these figures of and integrases, which are, of course, of the RNAi family. And so I kind of was looking at the Peewee domain a little interestingly, but Jejun did the final real test, which was already check on the database and found the similarity to these RNase H proteins down to the active site, down to the two critical aspartate residues that are invariant RNase H enzymes that sit in two adjacent beta strands. So we really excited about that and compared what we knew about this family of enzymes to what we knew from mostly Phil Zamor's work and Tom Tuchel's work about how the cuts target RNAs. And we realized that it was very similar, both magnesium dependent, same kind of products. And of course, you say, the saying goes, like a duck and quacks like a duck, it is a duck. And I'm sure most of you, or some of you, we believe that Argonaut looks like a duck. Here's the head of the duck, and here's the body and the little tail. And I can tell you now that IOEM, the 2D reconstructions really look like little ducks. Okay, so the space the RNA where it would cut also fit. We put in overlaid Dinshaw's structure with the RNA here, elongated the messenger RNA, and it was would cut at the right place. And then went down, showed Greg Hannon all of this, showed him which residues would be relevant, which aspartates would be relevant on human Argonaut 2 to mutate. He did that, and we can show that this really was the entity that was responsible for slicing in RISC. We later showed that apart from the two aspartates, there was a histidine involved. And then later on, Dave Bartel and Dinshaw Patel showed that there's actually a fourth residue involved in the active site. So what this told us was that Argonaut, apart from being slicer, was binding to the guide was really at the heart of risk, binding to the guide at the 3' end, and then directing the complex to the target through hybridization. When it slices, it positions the target at the right place in the active site for cleavage. It also told us that this was a programmable enzyme, and that really is the feature that in for therapeutics and for tools and what revolution before CRISPR. And of course, we know that CRISPR has the same kind of philosophy of being able to swap in specificity you dial in. So 8 years after that, to get a structure of a bona fide human or mammalian, and in this case, a human Argonaut, it was published by Ian McCrae in Science. What they did was to crystallize human AGO2 with RNA that came along during the expression of the enzyme. So it was crystallized with a heterogeneous sort of pileup of RNAs in it. We were trying to also do this, except fish out Argonaut without the RNA that was bound to it. And Elad, when he came to the lab, managed to do that, managed to separate a fraction of Argonaut that didn't have RNA bound to it, and then could add in RNA and then crystallize with a particular RNA. In this case, it's miR20A. It looks very similar to all the other ones. We had other eubacterial ones already, and then here Argonaut has already a little beak. One feature that I'm not going to go through all the features, but one feature that I did want to mention is that Argonaut with RNA in it, whether you bring it in from the expression system, is really stable. You hit it with proteases and it doesn't really touch it very much. If you take the RNA out, it looks like any other limited proteolysis experiments, cuts it in. And when you add back the RNA, it gets back to being very stable. The reason for that is that the RNA seems to basically tape all of the domains of Argonaut together, very stable structure that's and that really causes like symbiosis between the protein and the RNAs that are bound. One stabilizes the other. So the microRNA stabilizes AGO, the microRNAs in the cell. Skip that. And just mention these, again, these two pathways that you know about. When there's complementarity, it cuts. When there's less complementarity, then it recruits other factors that eventually destroy the mRNA target and also inhibit translation. But I want sort of end to telling you a little recent story that's not published, and just thinking about how this regulator of genes, Argonaut, is itself regulated. We know that it undergoes a whole bunch of post-translational modification, ubiquitination, phosphorylation. One of these was described by Josh Mendel and also Günter Meister's labs. And this is phosphorylation on the eukaryotic insertion. And this is phosphorylation by CDK1 and then dephosphorylation by a phosphatase called PPC6. It suggests this insertion is present in Argonauts, not in prokaryotic ones, and also not in the pewees. All of the Argonauts, and I'll talk about that in a little bit later. So as I mentioned, it gets phosphorylated by CK1 alpha. It gets dephosphorylated by PP6, and it looks like a cycle is actually important, because if you use either a phosphomimetic these residues or mutate out these phosphorylatable residues to alanine, you don't get the same kind of regulation. You actually dysregulate these mRNA targets. And so here, Bree, who's a graduate student about to graduate in the lab, you may recognize her from her posts as the bumbling biochemist, big following on Instagram. I think it's like 30,000, like that. She was interested in studying how this all works and started by looking at a guide target pair. And because we were able to easily produce Argonaut 2 without any RNA in it, she showed that this insertion does not get phosphorylated without any RNA. If you add guide, it still has no phosphorylation to see. But once you add the target, then you really see a really bump up in phosphorylation of that region. We know it's on that region because if you take an Argonaut with mutated sites, it can't phosphorylate it at least with CK1 alpha. And so then seeing what properties of this target are important for this phosphorylation, and used a whole array of targets and found that you need at least 14 a length of a target in order to get this phosphorylation. Then she looked to see if what other features are needed, and saw that you actually need some hybridization in this supplemental region. So this is not fully complementary, but there's some base pairing beyond where the seed sequence is. And you can see that when you have that, you have a bump in phosphorylation, whereas if you have only seed sequence match, it's low. But if complementary RNA, in fact, you do not see this kind of phosphorylation. So you really need this supplementary, extended supplementary region, but not fully complementary. And that is the case whether it's a Argonaut that can slice as in the wild type, or if you mutate out these active site residues. We know that from Ian McRae's work that these different targets do is in how the Argonaut is another experiment she did was to look at how phosphorylation might promote target release. And you can see that when we have phosphorylation, the target is removed very quickly from Argonaut, whereas when you don't, it doesn't go very quickly. And then again, that is dependent on the eukaryotic insertion, because when you mutate those phosphorylatable residues, you don't see this effect. So, and this is kind of an additive effect. The more open sites that are phosphorylatable, the higher the off rate of the target from Argonaut. And so, so just to kind of wrap, when you have target binding, that's when you see phosphorylation from CK1 alpha. And in fact, Bree showed that there's an order to this phosphorylation, a particular order. And then once you have these phosphorylations, target can get released, and then this can be recycled into, once the phosphatase comes in, erases those phosphorylations, and now we're able to bind target again, and then that induces, and so on and so forth. And then just to mention here that true for the Argonauts, these residues are conserved for the Argonauts that deal with microRNA. But for example, the fly AGO2 that does not, that deals with siRNAs opposing to AGO1 in flies, these residues are not there. And others with microRNAs, they don't retain these phosphorylatable residues. And so, so I'll just actually skip this again. This is just kind of wrapping up. And to thank all the people that were involved, people in the work, Elad, and then Bree with help with some very capable people in the lab. And thank you.
Thank you, Limor. That was fascinating. Couple questions here from the audience. Actually, first one from Craig. Do you think, given the modularity, that AGO2 could be split and the two parts assemble and function in vivo? Not sure if that's been tried. What do you think there?
If Argonaut itself can be split and function?
That's what Craig asked. If AGO2 could be split and the two parts assemble and function.
I don't know. I mean, I don't know if anybody tried that.
Right.
That'd be kind of an interesting experiment. Maybe it could be stitched together with the RNA. I would imagine that would be able to work together, but I don't know. That's a kind of an interesting experiment.
Yeah, I suspect the RNA could stitch together.
Yeah.
That'd be worth. Another question here is, could you please comment more on how AGO structurally distinguishes an sRNA versus a microRNA?
Yeah, so I think we're not quite there yet. The residues around where it's supposed to cleave are really important in how they change how Argonaut is organized, and there's some from Ian McRae's lab that also shows that. We've been actually looking now with hydrogen exchange and mass spec hydrogen exchange to really look at these transitions basically in solution, and basically allowing kind of these transitional states with the different types of RNA. And you can see that there's when you go from completely RNA free to adding guide, and then when you add different targets, the transitions or the differences are a lot more subtle. But we also know that there's other residues that are not far from the active site that also have something to do with sensing whether it's completely complementary or not. But I can't say that we know the full answer to that yet.
Okay, thank you. And then maybe one last question, Limor, why is position 10 in the guide strand so well tolerated for chemical modifications? Is that region clearly studied for AGO2 structure?
No, not really. I mean, it's much more open. So T2a sequence, the RNA binding region is a lot more restricted. It really fits just so, and it's open to interacting with the target, but not a lot of room. And then it kinks around residue 7. And after that, it becomes a lot more open. So there is a lot more room after that to add. Exactly position 10, we really didn't look into it that carefully.
Okay, thank you so much.
Sure.
All right. I think we're going to now transition to the final talk, which is my talk during the session. And let me just see if I can get this moving the right way. I believe you can see my screen. To participate in the session. I'm John Merganori. I'm the CEO of Alnylam Pharmaceuticals, and have had leading the Alnylam team for the last 20 years, virtually 20. So let's proceed. I think this slide doesn't need much of an introduction, but it just, the foundation for what I will talk about. I mean, when we got started back in 2002, it was really based on the vision that the wonderful research and findings that both Craig and Limor talked about could be harnessed with the goal of targeting specific mRNA that encode mRNAs that encode proteins that are involved in the cause and pathway of human disease, and target those mRNAs for silencing so that we can shut down the production of the disease-causing protein, a very simple idea in some ways, and yet a very complicated endeavor in many others. Now, these are the data that certainly are what we're celebrating today. The work of El-Bashir et al. from the Tushel lab, obviously so inspired by Craig's work and many others as well. Phil Zamore, many others. But of course, these were also the data that inspired a group of venture capitalists and myself to work with scientists from many institutions, including UMass, T. Whitehead, Max Planck, to start this endeavor of medicines based on the discovery of RNA interference. And I think these data are very notable by the silencing of the lamin gene. Where that's led us, and I'll highlight as I go into my talk, is an approach that allowed us to figure out how to make drugs out of siRNAs and specifically how to achieve delivery of small interfering RNAs into initially hepatocytes, and we believe in the future other cells in the body, and to build what has become a clinical pipeline of over a dozen programs from phase 1 to phase 4 clinical development, with programs that are both rare diseases and also prevalent diseases. And this is the work that has in our hands over the last 19 years. Now, notably, we've now brought 4 therapeutics to markets around the world. And this began in August of 2018, a day I'll never forget, with Onpattro's approval for the treatment of the polyneuropathy of hereditary Attr amyloidosis. And then a year later, the approval of the first GalNAc conjugate siRNA for the treatment of a rare disease called acute hepatic porphyria. And then about another year later, the approval of a third RNAi therapeutic, Oxlumo, for the treatment of primary hyperoxaluria type 1, which is the first pediatric approved RNAi therapy. And then while FIO, which is targets PCSK9, awaits its approval in the United States, it markets around the world, including Europe. And this is the first RNAi therapeutic approved for, namely, the treatment of hypercholesterolemia and mixed dyslipidemia. So the beginnings of the RNAi therapeutic programs that have really been brought ultimately to markets around the world and to really make a difference in patients' lives. But it's in a little bit, and just taking Wall Street estimates for research analysts that work in Wall Street of where they expect additional approvals to occur from the pipelines of Srna and Arrowhead and Silence, you can see that over the next 5 years, it is estimated that we could have about 20 RNAi therapies that have to regulatory approval. This isn't risk adjusted, so may not get quite to 20, but it is really an impressive growth that we can expect to see over the 5 years. And obviously much more beyond that will almost certainly occur decades to come. So this is a wonderful illustration of just how the basic discoveries of RNA interference truly materializing into a whole new class of medicines that can greatly benefit patients and with a broad range of human disease for many years to come. So what I'd like to do is really focus on 3 topics, illustrate the progress on RNA interference, rare diseases, then transition to common diseases, and then talk a little bit at the end about some new frontiers of interest to us at Alnylam. And I'll start with ATTR amyloidosis. This is a progressive disease that is ultimately debilitating and for patients, that is caused by misfolding of the transthyretin protein expressed in the liver. and the resulting accumulation misfolded protein fibrils as amyloid deposits in a range of peripheral tissue result in tissue pathology. And as you likely know, there's a hereditary form, 50,000 patients worldwide, and then there's a sporadic wild-type form of the disease without a known etiology why wild-type TTR can misfold over time, but that affects a much larger group of around the world. Now, the detailed mechanism has really been well defined. A lot of this work comes from the work of Jeff Kelly and also Merrill Benson, who sadly just passed away. But you can see on the top the very clear delineation of the pathogenic mechanism of ATTR amyloidosis, starting with the expression of the TTR protein in the liver, its propensity to misfold either via mutation or otherwise, amyloid deposits in tissues. And what you see on the bottom are the wide range of interventional strategies that have emerged over time, some that have been still investigational, but I think you can appreciate that there have emerged multiple ways to target this disease, but certainly RNA interference as well as antisense oligonucleotides represent two examples of approaches that have reached the market. Alnylam, we've really brought forward three different programs. One is approved, that's Onpattro. That is a lipid nanoparticle encapsulated small interfering RNA, the only program with a lipid nanoparticle that we took forward. We're all very fortunate that we did this work on lipid nanoparticles, even though it only led to one RNAi therapeutic, but certainly the work that we did on novel lipids really was the foundation for the lipid nanoparticles used in all of the mRNA vaccines that many of us received over the last year. For that contribution, not only for Onpattro, but also for the broader public health objective. Vutrisiran is I'll spend a little bit more time on vutrisiran. This is investigational. It's a GalNAc conjugate, and it has some promising phase 3 data and is now the FDA and the EMA for potential approval, which, if positive, could result in the fifth RNAi therapy to get to market. And then finally, a very development that I'll talk about at the end of my talk, using a new platform that we developed for being a much more longer lasting RNAi therapeutic effect that we believe will be almost like a vaccine for treating disease. So let me talk about vutrisiran. I've heard about Onpattro before. I don't think we need to go into that story very much, but on the phase 3 results with vutrisiran that were presented earlier this year. The phase study was really a very innovative design. It was a randomized open-label study where we vutrisiran against the historical placebo group from the original Apollo study. That was done because it really was not considered ethical to randomize patients to placebo for treating hATTR polyneuropathy. And this study had a 9-month efficacy endpoint using a neuropathy impairment score called mNIS+7, and it also had a number of other important secondary and exploratory endpoints that were investigated. So this trial, we were really happy earlier this year to show positive results for the study. This is a summary of those results here on the slide. What you see on the left is neuropathy impairment score. This is a multi-component score that's done by a neurologist to basically measure the progressive impairment of neuropathy in patients. And what you see in red is the historical placebo increase or worsening of neuropathy in patients that receive placebo over the 9-month period. And then in the blue line, you can see the benefits of vutrisiran administration, which is given as a once every quarter subcutaneous injection, low treatment burden for patients, 4 injections a year to generate, the type of results that we achieve with Onpattro, which is given by intravenous infusion. So we think that these results can potentially, if approved, lead to a medicine that really provide an unprecedented level of freedom for patients from their disease. And then you can see the categorical binary analysis that's done to evaluate the frequency of patients that actually have a lower mNIS+7 score at 9 months baseline, indicating evidence for actual improvement of their disease. And over the majority of patients show some evidence for improvement. On safety, the results were really very encouraging. The safety findings were generally with the disease and were actually trended lower in the treatment group relative to the historical placebo. And the only adverse seems to be related to therapy in this study were mild injection site reactions. So those have led to a submission for a medicine that we hope will be our 5th RNAi therapy that we bring to market. But we're spending a lot of time now in the ATTR space focusing on this other manifestation of the disease, from hereditary disease into wild type disease. And we're doing randomized clinical studies right now, one called Apollo B, which has over 300 patients that have been enrolled in the study. And we should have results next study, and that'll evaluate the safety and efficacy of patisiran, which is the non-proprietary name in Onpattro, for the treatment of both hereditary and wild type ATTR cardiomyopathy. And so we look forward to those results next year. And then the Helios B study is a much larger study where we're looking at 30 months of treatment. And in this case, we're looking at vutrisiran. We also have completed enrollment in this clinical study with over 600 patients, and we should have data in early 2024 and potentially earlier from analysis. Now, there's good reason to be encouraged by an RNAi silencing mechanism of action in patients with ATTR cardiomyopathy. And this comes out of some exploratory data from the original Apollo study highlighted in the upper left of this slide, including improvements of cardiac biomarkers like NT-proBNP, improvements in echocardiographic measurements as well. And then importantly, on the bottom left, a safety profile, safety perspective, which has also been encouraging. But more recently, and this is highlighted on the right-hand side of the slide, some out of the National Amyloidosis Center in the UK really highlight some of the unique features of an RNAi silencing mechanism of action. And here on the upper part of the right side, you can see the benefit of patisiran on cardiac amyloid detection after 1 year of treatment in this single patient here, where you see evidence of regression of amyloid deposition in the heart of these patients. And then on the bottom, you can see some correlates of that effect as it relates to extracellular volume and 6-minute walk distance, and then also NT-proBNP. So these data really do around the potential of an RNAi therapeutic approach of ATTR cardiomyopathy. But of course, we await the results of randomized of the potential safety and benefit. So let me now turn to an area of interest to us, which is really to focus on the broader number of patients that have disease where RNAi be impactful. And the cartoon illustrating our journey as we have been in RNAi-based therapies, initially in rare diseases, monogenic, where the unmet need is very high. It's obviously a very good place to get started. Modality like RNA interference, but over time we migrating to the more prevalent disease settings. Of course, this started with Leqvio and inclisiran and PCSK9, but more and more programs at Alnylam are focused on these opportunities. And amongst the many reasons that we believe this is important and a good place to explore are the facts that these remarkable durability. I mean, we've seen quarterly administration being possible every 6 months, dose administration being possible, minute annual dose administration being possible as well. And when you can get so infrequently as once a quarter, once every 6 months, once a year, you really have a public health issues, largely because of the fact that both a clamped pharmacodynamic effect on interest, but you also address one of the most important problems in medicine today, which is patient compliance with therapy. And a drug that can be given patients' visit to their doctor is something which really can improve adherence to medicines that ultimately are needed for life-saving benefits. And so that's really an important opportunity that we think is going to be inherent mechanism of action. So with that background, of very recent interest to us at Alnylam has been how RNAi therapies help us reimagine the treatment of hypertension. Hypertension is the leading cause of CV morbidity and mortality around the world, and it's a public health crisis due again to the lack of patient adherence to medicine, but also due to the fact that existing antihypertensive drugs don't generate lowering of blood pressure. Patients have sawtooth pharmacology of their medicines that they take, assuming that they take them compliantly, and that sawtooth creates the lack of tonic control of blood pressure, which is associated with clinical benefits in terms of cardiovascular risk. And so the ability and clamped pharmacology of an RNAi therapy combined with the benefits really do allow us to think differently about how an RNAi-based strategy in a very prevalent disease can potentially allow for a reimagining of this condition. So the approach that we are taking is to target the hepatic synthesis of angiotensinogen. Angiotensinogen, of course, is the substrate precursor protein for vasoactive angiotensin peptide. It is expressed not only in the liver but also in the kidney. And having a liver-selective knockdown of angiotensinogen is believed to be able to provide control of blood pressure without the adverse effects that can be seen with other RAS inhibitors, kidney function, which may be due to the targeting of angiotensinogen in the kidney. And so this liver trophism for an RNAi-based therapy is, in fact, a desired approach creating effective and potentially safe interventions for hypertension. And these are the phase 1 data that we've recently presented. And on the left-hand side, you can see the dose down of angiotensinogen that is obtained from doses as low as 10 milligrams in the study to as high as 800 milligrams in the study. And you can also see just the remarkable durability of knockdown effect that we enjoy with these chemically modified sRNAs. And you can see just a single injection at time 0 at the highest dose groups are generating knockdown effects that are exceeding 80% for nearly at least half a year. And this is why we are encouraged by the potential for infrequent once a quarter or once every 6 months. And then on the right, you can see that angiotensinogen silencing on blood pressure. And at the 3 highest dose groups, a bit of a plateau. At those dose groups, you can see systolic blood pressure reductions exceeding 10 millimeters, and at the highest dose group exceeding 15 millimeter mercury reduction as monotherapy. This is without any additional antihypertensive medicine. And all these effects are placebo controlled. And it's also been encouraging to see really a very encouraging ability profile with no evidence for any hypotension requiring intervention, safety profile, again, injection site reactions occurring as is common with drugs. So with these data, we've now started a very robust phase 2 program that'll over 1,200 patients. And the first study has been kicked off and the second study starting shortly thereafter. So look forward to having perhaps the first study at the end of next year. And these are the studies that I just described. Cardia 1 as monotherapy, and Cardia 2 is being done as add-on therapy with additional antihypertensive drugs, including RAS and calcium channel blockers or diuretics. And the Cardia 2 study will start relatively shortly. So I want to just close with on a few areas of recent interest, or actually one in particular of recent interest that I wanted to share some new unpublished research from our laboratories. And I'll start by just saying that one of the things that we've been very committed to at Alnylam over the years is continuing to invest in our platform. And we've done a lot of work in delivery of RNAi therapies. We have our first CNS program that should go into the clinic this year. That'll be an important milestone because just like we've done with the liver, where we've opened up the ability to reproducibly and in a highly modular hepatocyte expressed target genes, we believe we can do the same thing in the CNS. And again, hopefully sometime this meeting, perhaps next year, we'll be able to talk about the first human data from targeting a CNS gene, a precursor protein. But the other other areas of interest are highlighted here as well. Make strong progress across our whole platform. But the topic du jour that I wanted to share really comes to the continued enhancement of our GalNAc conjugate platform to achieve even better pharmacodynamics, and a new version of our platform that we're now calling Icaria, which is an island in Greece, a country I love dearly, where it's one of these blue zone islands and people live for a very, very long period of time. And so these are long-lived days that we hope will make a difference in long living of humans at the end of the day. And I think people appreciate that the evolution of our platform for sRNA targeting really has changed quite a bit over time. And you can see how our original standard template chemistry platform required high doses and also was relatively short-lived compared to our ESC+ platform programs with much longer durability, as you can see on the right. And these are the identical sequences, but really this just shows the benefits of different chemistry that we've used, backbone as well as nucleotide chemistry as well. Now, the benefits of pharmacology is really, again, exemplified from the work that was done by the Medicines Company and now taken forward by Novartis with inclisiran, which is approved in many countries around the world and still under review in the US. And here, a once every 6 monthly injection generated an over 50% reduction of LDL cholesterol with a remarkably encouraging safety profile. Again, injection site reactions were the most common adverse event that were all mild and generally well tolerated. But we've learned a lot about this remarkable durability. And I some of the data that, in fact, has been featured as the paper of the year here at OTS, a paper that was published by Chris Brown and colleagues at Alnylam. And using endolytic peptides, what they were able to show is that administration of the endolytic peptides at different time points after the administration of the sRNA resulted in a burst of RNAi silencing activity, and the fact that the durability of these sRNAs is really linked to a depot that exists intracellularly. And there's continued work at Alnylam to really delineate this mechanism further, but it really is the depot effect that is the highly durable clinical effects that we see in human studies, but also see in preclinical studies as well. So with that insight in mind, this Icaria platform that we brought forward is something that we're very, and really at the heart of it is what's shown here on the left. We've been able to really engineer sequence and chemistry to achieve exquisitely specific sRNA molecules where the on-target effect preserved, but the off-target effect is really completely neutralized. And in the middle, you can see how a molecule like TTR-SCO4, which is our sentinel program of this platform, compares to an analog of vutrisiran. This is the molecule that is currently under review by the FDA and supports quarterly, if not biannual dosing. And you can see that TTR-SCO4 is equipotent, if not a little bit more potent than this vutrisiran analog. And when we model these pharmacologic effects, and our modeling has become very strong and very good, we can clearly see the ability of having a once annual dosing regimen with target knockdown that exceeds well over 90%. And of course, this is enabled by the improvement of specificity and the ability of going to higher doses. Instead of using a 25 or 50 milligram dose of vutrisiran, we can use a higher dose of TTR-SCO4 to achieve this very durable annual with, again, a deeper level of knockdown. So we think this is an approach that will be enjoyed in other programs at Alnylam, but will allow us to really think about vaccine-like benefits in these things in a way that we think can be revolutionary for medicine for many years to come. So I want to close with this slide. I really have enjoyed being part of this journey. Craig, thanks to you, Limor, your work as well. We got started in 2002, 20 years ago, around the time of the discovery of RNAi in mammalian cells. I got started in this field at that same time. And the journey that we've been on has really been tremendous and exciting, and something which I think all of us really are happy to have been part of. And I will end my talk here, and we can go to Q&A if there are questions. So I'll stop sharing. Good. So okay, let's see. Question here is, thank you for sharing this. What mRNA is targeted in ATT? Ask if it is not a secret. No, it's not a secret at all. It's transthyretin. It's the transthyretin that is expressed in that's what we target for the ATTR program. Do you see sRNA disease prevention or prophylactics? Absolutely. Again, as I just a once annual sRNA approach through this IKARIA platform, we do believe, like, in terms of its ability to prevent diseases that are major public health crises, such as diabetes or metabolic syndrome or dyslipidemias, hypertension. We believe that we can engineer these molecules to effectively be vaccine-like approaches to these very prevalent diseases. And maybe I'll take one last question. What is the administration of sRNA drugs? Well, it depends, right? At one extreme, you have molecules like patisiran that are given intravenously every 3 weeks, and obviously have done very well to help patients. And then at the other extreme, in the clinic, we have a once approach for inclisiran for the treatment of LDL cholesterol, reducing. We hope eventually to get to once annual with a and then so it is a range. The range is relevant oftentimes for what types of diseases for therapies. So I think that's where we should stop. But I do want to thank our speakers, and I do want to thank the audience participating in the session. I believe the meeting is now adjourned for the day, and everybody for participating. Thank you very much.